Hi everyone, my name is Julia Schilling and today is August 25th of 2022. In my presentation, I will be talking about the determination of gender, specifically looking at genes and hormones. To begin with, I think it is important to go over some vocabulary. A couple of terms that we need to establish begin with external genitalia. This includes all of the external reproductive organs. On the other hand, the internal genitalia is the organs, accessory glands, and ducts that are within the true pelvis. They connect the gonads with the outside world. Gonads are the organs that produce gametes. Gametes are the reproductive cell of humans. For males, the gonads are the testes that produce sperm. For females, the gonads are the ovaries that produce eggs. The female that produces the egg and the male that produces the sperm come together, and at first they are both haploid, but when they come together, they produce what is known as a zygote, and it is diploid. Both the mother and father give 22 pairs of autosomal chromosomes and 1 pair of sex chromosomes. The autosomal chromosomes directly contribute to the development of the body. They help with the development of things like hair color and blood type. The sex chromosomes determine the sex of the baby. They can be either XX or XY. A female can only give X chromosomes, but the male can pass on an X or Y chromosome. If the chromosomes are XX, it will be a female. If it is XY, it will be a male. In the image on the bottom right, you can see how the egg comes together with the sperm, and depending on the chromosome it is carrying, it will determine whether the individual will have XX or XY, which will be a zygote and it will be diploid. Next, we will get more into the process of sexual differentiation. This occurs early on in development, specifically around the 7th week. Prior to differentiation, the embryonic tissue is bipotential, meaning you can't identify the tissue as belonging to a male or female. Looking further into this, the bipotential gonad has both an outer cortex and an inner medulla. If there is the presence of the appropriate developmental signal, then the medulla will become the testes of a male. If there is an absence of this developmental signal, then the cortex becomes the ovarian tissue of a female. The bipotential internal genitalia consists of Wolffian ducts and Mullerian ducts. The importance of these ducts is that depending on whether the embryo is male or female, one of the ducts will continue to develop while the other degenerates. Looking at the bipotential external genitalia, it consists of the genital tubercle, urethral folds, urethral groove, and labioscrotal swellings. The external genitalia will develop into the external reproductive structures of the male and females. The image to the right here is showing how the bipotential gonads can develop differently due to the Wolffian and Mullerian ducts. If the Mullerian duct degrades, the Wolffian duct becomes the epididymis and vas deferens of the male. If the Wolffian duct degrades, the Mullerian duct becomes the uterine tube and the uterus of the female. Looking at female embryonic development, after the development of the ovaries in the female fetus, one of the X chromosomes becomes inactivated and condenses into a Barr body. A Barr body is a clump of nuclear X chromosome chromatin. Because of this, you then have either an active maternal X chromosome or an active paternal X chromosome. Continuing on, we will look at the SRY gene. This gene provides the instructions for making a protein called the sex-determining region Y protein. In the absence of this gene and the presence of multiple female-specific genes, the bipotential gonad, specifically the cortex, becomes ovarian tissue and the gonadal medulla regresses. Additionally, the absence of testicular AMH allows the Mullerian ducts to develop into the upper portion of the vagina, uterus, and fallopian tubes. With this, the Wolffian ducts degenerate due to the absence of testosterone. Then the absence of DHT will cause the external genitalia to acquire female characteristics. We can now look at male embryonic development. It is the presence of the SRY gene, which was mentioned in the female embryonic development, that allows for the development of the gonadal medulla into the testes. With this, the gonadal cortex then regresses. It is important to note that testicular development does not require male sex hormones like testosterone. This is because testosterone cannot be secreted until the gonads differentiate into testes. As mentioned earlier, the SRY gene produces the testes-determining SRY protein. This binds to the DNA and activates the genes SOX9, WT1, and SF1. It is the testes-determining that produced by the SRY gene that causes the gonadal medulla to develop into the testes. In the image on the right, this pathway can be seen visually. Once the testes develop, they contain two types of cells. First, they have interstitial cells which secrete testosterone. The secretion of testosterone causes the Wolffian ducts to develop into accessory structures like the epididymis, vas deferens, and seminal vesicle. Additionally, it causes the development of the male external genitalia. The other type of cell is Sertoli cells. These cells secrete anti-Mullerian hormone, causing the regression of the Mullerian duct. Here on this slide, I have included an image showing how the SRY gene controls testicular and ovarian development. It shows how the addition of the SRY gene causes the production of 3 hormones and how it changes the gonadal medulla into the testes, while the absence of the SRY gene causes the production of hormones that help to form the ovaries from the gonadal cortex. Here is the reference I used for all of my information. Thank you for listening.